I thought I'd come up with a slightly provocative title, and really to just demonstrate where we do translational research and the way we think about translational research and how we use all the different tools, methods at our disposal to address key clinical and biological questions. First off, I'd like to say that it is a pleasure and a distinct privilege to be here, also to see so many smiling Glaswegian faces, and also Jason Carroll, a fellow Australian. We in fact did our PhD together. He did finish 1 year ahead of me. He did teach me how to do my first protein extraction, and I've never admitted this, but it was so complicated I just went and bought a kit to do it the next day. So we think of translational research as a mindset, not a method. What tools do we have available to us to address these particular questions, and trying to be as close as the clinic as we can? And this is the Wilson Wall Cancer Research Centre that I direct, and it's really focused on translational research. And what I want to talk about today is really some of those early things and then our road to precision oncology and some of the challenges that we face. And I will suggest that precision medicine is not a new paradigm shift, it is just the natural evolution of healthcare. And where our biggest challenges lie are not in science, are in implementation and operationalization, if there is such a word. For those of you who don't know where the pancreas is, that is it behind the stomach with a dirty big lump in the middle of it, which is a pancreatic cancer. It means you can eat all of it in Greek, pancreas. I don't know why they called it that. In fact, it's been neglected for many years. Many languages just call it that thing under the stomach. This is what the pancreas looks like during an operation. And this is one of my first early surgical mentors who took his left, while I was a registrar, his left hand with his stubby fingers, put them around the pancreas and said, "Yes, we can take this one out." And I asked him the question, well, how do you know that? Well, experience, my dear boy, experience. And there's nothing more sensitive than the fingers of the left hand of a pancreatic surgeon. Then I pointed out to him that we now have these things called CT scans, so you can actually see this bit. There was a bit of a humph. And then I went on to electron microscopy, and at that stage I was asked to leave. Just in case you're interested what it really looks like, this is what it looks like after you take the pancreas out. The veins are blue, arteries are red, the gallbladder is sort of greenish, and this bit's just a little bit of residual blood. And that's fat, for the people that want to know that. This is what the pancreas looks like microscopically. There are two components involved in digestion. This is the exocrine pancreas, which secretes all those enzymes that help you digest the steaks, etc., that you eat, or if you're a vegetarian, the vegetables. And this is the endocrine component that has insulin and other hormones that regulate. Endocrine secretes into the body, which is the blood, and exocrine, in fact, the gut. To my surprise, in final year medical school, I realized it was actually outside your body, not inside your body. So pancreatic cancer. Pancreatic cancer is the cancer of this component, the exocrine component. This is slightly different. This is what Steve Jobs died of, and this is what Patrick Swayze died of. So we're talking about the Patrick Swayze type of cancer, which is tremendously aggressive, just overtook breast cancer as the cause of death in the United States. Less than 5% alive at 5 years. 90% die within a year of diagnosis. Hardly anything's changed for 50 years, and the average survival is 6 months. Many are diagnosed at autopsy. We've come a long way in the way we develop cancer therapeutics. We've gone from fairly broad generic mechanisms in chemotherapy, surgery, radiotherapy, through to targeted therapies, really based on our understanding of molecular mechanisms. This is just by way of example, the retinoic acid signaling pathway, identifying where you can intervene in this pathway. And so we've moved from a more generic screening approach to understanding the biology. And this has been tremendous in our transformation of how we look at cancer and treat cancer, but it's also caused us particular challenges and problems. Many suggest that it's only the last decade or two that we've been doing targeted therapies, but in fact, probably the first example of targeted therapy was George Beatson in 1896, who removed the ovaries to treat breast cancer. He managed to get some case reports published in The Lancet. I've sent them a few and never had any joy. Again, we can use different mechanisms, and this is interesting. This is probably the most successful targeted systemic therapy ever, tamoxifen. And it was sitting on ICI's shelf for years. I can't remember what it was developed for, until once clever cookie said, well, wonder if it actually targets estrogen. And this is, for those who don't recognize it, is the perineum of a baboon in heat. And you can actually, the experiment that was done was to give the baboon tamoxifen, and it suppressed that particular perineal flush. And now tamoxifen and antiestrogens that evolved from that are the most successful therapies for targeted therapies for breast cancer. We also use other models, and the realization that cancer shared a lot with embryonic development was really important in the way we started to develop therapeutics and understand mechanisms. For example, this is the pancreas developing, a couple of buds from the endoderm all the way into this beautiful structure of the pancreas we saw previously. And we see this as carefully orchestrated by a number of mechanisms, some of which you'll see as familiar, that we have drugs towards hedgehog, drugs towards notch, we have drugs towards retinoic acid. And this carefully orchestrated events allows us to develop our gut, our pancreas, and other things. And in fact, to my delight, I learned that the pancreas is the default mechanism of embryonic development in the abdomen. If you knock out hedgehog, you end up with one big, massive pancreas. Not particularly useful, but it was really satisfying to hear that. If we go back to the origin of the bug, so this is the bug, this is the Drosophila, and I wish I was around in the day when we started describing names for genes. So this is hedgehog, and you can see this is the normal larvae, and this is cool, look like a hedgehog. Plenty of other inventive names like disheveled, etc. Maybe those guys should have got out a bit more. The other things that we use to study, and this is a great model, is sometimes you need to reproduce things in multiple different ways, and you can't really do that in mice or other systems. And so this is the zebrafish, and I still am the only surgeon that's operated on zebrafish embryos in Australia. They can produce thousands of eggs, and here's the blastocyst at the 2-cell stage, and simply by injecting this a little bit of whatever into here, morpholino antisense oligonucleotides or capped mRNA, you can start to influence these particular fish. And this is rhodamine dye being taken up by the zebrafish embryo. And in fact, the zebrafish does have a pancreas, but one single islet. And by manipulating this is 80 hours post-fertilization, either through knockout animals, to knockout genes, or injecting them, you can start to understand and unravel the mechanism of these pathways in the embryo, which are informative in what we do in cancer. And this is work that I did while I was at Hopkins. We can always use mice, and if you think taking out a human pancreas is hard, taking an E11.5 pancreas out of an embryonic mouse is a little bit trickier. Thankfully, the medical indemnity is not as high. So things started to change, and it's often in science, technology can be a major driver. And one of the things that always fascinated me, and some of the work that I did at the Garvan, was really trying to understand the differences in response and the difference in prognosis for different cancer types. And all of a sudden, next generation sequencing came along. Went from the first genome, one genome took, I think, 15 years and $3 billion to the fact that we could start doing it in our own labs almost, only probably 100 million. Which is a lot less than 3 billion. We worked on pancreatic cancer, and we had the privilege of working with Sean Grimman to be able to sequence, as part of the International Cancer Genome Consortium, 24 to 28 countries, 90 projects, which evolved into the pan-cancer analysis of whole genomes, which is informing how we can start to develop therapeutics and better select patients, to the next iteration called the ICGC for Medicine, which focuses on clinical trials and other aspects. For those of you who haven't seen a cancer genome, this is what a cancer genome looks like. It's a Circos plot. It shows the chromosomes around the outside and the various different abnormalities, either point mutations listed here, copy number changes, and big chunks of DNA that move across, which are called structural variations, translocations or deletions, etc. And what we found that these genomes were very complex. And as you started to piece them together, finding recurrences and similarities was very difficult. But nonetheless, even in this and each one of these blue spots is a gene, and each column is an individual patient, we started having a lot of diversity, but we can group them based on either their genomic changes, which may reflect an underlying process, mutational signatures, which represent exposure like tobacco, ultraviolet light, or old age, and also mechanistic pathways. And I won't go into these in detail, but this diversity was of particular concern. And when you, what have I done? Wrong button. And so when you look at this particular diversity and you look in for those targets where drugs have actually already been tested in pancreatic cancer in unselected ways, you see the actual molecular targets of these particular drugs. But it's little surprise that they failed, because if you have even a 5% prevalent subtype, you need about 30,000 patients to prove a difference. So if you can imagine you're doing a clinical trial of maybe 30 or 40 patients, and only 1 or 2 of your patients has that particular subtype, you're not going to detect that signal in whatever shape or form. And hence, the principle of precision medicine, precision oncology, matching the right drug to the right patient, seemed particularly important in pancreatic cancer. But it wasn't just the case for pancreatic cancer. We can see how we group things based on organ of origin and some morphological stratification. In fact, when we look genomically, there's a lot of diversity. And where we've had those successes with particular drugs is where we've had a dominant subtype, and we were powered well enough in our clinical trial to detect that difference when we saw targeting this particular c-KIT mutation or targeting BRAF mutations or even targeting EGFR mutations in that context. And of course the estrogen receptor here, so you're really powered well enough in a smaller clinical trial that's feasible to be able to detect those particular differences. And so we saw a lot of these targeted agents failing, and lots of challenges for all of us as a community because the cost of therapeutic development are transferred onto the community whether we like it or not. And so there are challenges that we're faced. But nonetheless, when you get it right, even by accident, in this case, this is the pancreatic cancer. This is the tumour here that completely shrunk. This is a trace of a biomarker called CA19-9, and the height represents how much disease is present. This patient, just through sheer luck, received a platinum-based agent, had a deficiency in DNA damage response repair. Platinum's thought to target that in some shape or form, and this patient, who was 2 months away from death, lived for 4.5 years. And so that's by understanding the particular genome, the underlying pattern, and then matching the right treatment to the right patients. One of the other things we did, and this will become relevant later, is we generated cell lines and xenografts from all of the cancers that we actually sequenced. So then we could start to do experiments and understand and test these hypotheses in these particular experiments. One of the other challenges that we faced is that at the time we were getting these results back, these were patients of mine, patients of our practice, patients of colleagues of mine, I started seeing results and I thought, gosh, if that was me, I'd want to know. BRCA mutations, BRCA mutations, germline events that predispose to cancer. I really want to know that. And we had to amend all of our ethics to be able to return research results to patients. We put in 18 ethical approvals. Every one of them wanted an ethical defensible plan, and when you hear that, they don't know exactly what they want. And in the end, we ended up with 18 completely different ethically defensible plans. And that leads on to my challenges of how we institute precision medicine. We set up a clinical trial. This took us years to set up. We initially planned it in about 2010. It didn't actually start till about 2012, 2013. We could do the sequencing, we could get the information back in time. We couldn't sometimes get the tissue from the lab in time, sometimes months. And then we broke that by basically bribing pathology departments by saying, we'll give you $300 if you get the sample to us within 3 days. And this is probably the fastest paper I ever got studied and published, and some of the most recognition I got. It was submitted by a rather independent medical oncologist student called Lorraine Chantrell, who submitted it without my knowledge, and it got published straight away. And so that's probably a lesson for me and other supervisors in the room. So I'll get on to the next step. This is based on our discussion today. My supervisor, also Jason Carroll's supervisor, and our mentor, Rob Sutherland. And why he's up here today is that he developed pancreatic cancer during our sequencing project. And part of what we did in this context is we did everything we could. We took the sample, we grew cell lines, we grew xenografts, we tested it with drugs, we did all of those different things. We sequenced the hell out of it to identify what might be the best particular therapeutic. And this is around about 2012, and that sort of led on to something that we got involved in a little bit, which is sort of VIP oncology, which is you sequence samples for the rich and famous around the world, and you learn exactly what's happening in that particular cancer, and you might form a bit of advice. Now, that seemed to us initially as a path forward of how we can develop this, but the problem with that is that you just couldn't scale that in any particular form. And so in response to that, having moved to the UK, we're building platforms, and the platform we're building for pancreatic cancer is called Precision Panc. We built a similar platform called Precision Promise in the United States. We're working with the Koreans as well as the Italians and the Spaniards to really build out this particular platform. And this is really a labour of love, bringing together the NHS, industry partners, various institutions, etc., to really bring together that platform and allow us to really take the next step of where we need to move with pancreatic cancer in particular. And it's based on 3 pillars: discovery, preclinical development, and clinical development. And Owen is in the audience and also is here today as a new fellow, is a co-conspirator in this program itself. And really the principle is to align all of these different things, to align them in a way where you're doing similar experiments. And I noticed a document out there about reproducibility, to be able to reproduce things and really understand the interface. How do you prioritize your strategies? How do you build your preclinical platform of evidence? Taking what we know from our pharmaceutical colleagues and we work closely with, what are the key steps to take us to early clinical development and later stage clinical development? And then how do we continue to build a knowledge bank through taking deep science or having deep science as part of each clinical trial, and breaking through this particular diversity of targets that exists, but without a particular selection marker and matching the right patient to the right treatment, you're really not going to see a signal in that particular context. And so the clinical trial platform we built was really also very patient-centric. And this is about finding the trial for the patient rather than the other way around, which we've done traditionally. And really, in 2 steps, taking a sample. In precision medicine, you need to sample something. Doesn't matter what it is, you need to sample. You need to do a test that is going to give you some sort of information. You need to return that result in a meaningful time frame, and you need to have some sort of output for that. So you're actually going to, as a clinician, you're going to do something different because of that particular result. And so building this platform, the initial recruitment, and then providing a suite of studies, either built internally or through collaboration, to start to build out a knowledge bank and also to offer a trial for each and every patient in that particular context. We can do the science aspect of things. So we spent a lot of time building in Glasgow Precision Oncology Laboratory, building out exactly what we need to do to the scientific aspect, to do the measurements, even the discovery component, as well as the patient allocation component. But again, our challenge is really the clinical side of things, the network, the shifting, the established pathways in the clinic so that patients are considered for clinical trials because they may well be the right options. We have managed through proof of concept to be able to deliver these genomes, but scaling is probably the biggest challenge. And what I'll finish with, this is my last slide, is that clinical trials are now real treatment options for patients. Whether it's the Keytruda advertising by Merck in the United States, etc., but clinical trials are viewed as real treatment options, and this adds significant complexity into the way we practice medicine. And so we need to adjust our systems to be able to deal with that. Thank you very much.